Okay, so today we are talking about antibodies, and antibodies are made by your B cells, and each B cell makes a certain type of an antibody. They have the same basic structure, but depending on the parts of the DNA that code for different portions of the antibody, you're going to get different amino acid sequences, and that's going to make it so that you have different shapes on the end of the antibody that allow antibodies to attach to antigens. So the antigen is the foreign particle that your body recognizes and that your antibodies are then going to attach to. So they all start with the same basic structure, and it's in the shape of a Y, and the Y shape is made up of two what we call heavy chains. And so I'm going to start by drawing my heavy chain Y structure, and you should draw this in your notebook. I'm going to just draw it on this whiteboard. But it looks like this. So this is showing my two heavy chains of my antibody. There's also two light chains. So I'm going to draw the light chains. They're on the outside. And I am drawing them thinner just to kind of give you the idea that one is a light chain and one is a heavy chain, but they're not actually thinner and thicker. So that's my basic structure. And these things are held together by disulfide bonds. And if you remember back to when we did our tuber lab, we used those green push pins, and the green push pins could link together because they both had sulfur and they could form a disulfide bond. There are two disulfide bonds holding the heavy chains together, like that. And then there's one disulfide bond for each heavy and light chain combo, and it looks like that. So that's our basic structure of an antibody. But antibodies come in all different types, and there's one part of the antibody that's going to be the same for every antibody for a species, and that's this sort of bottom region here. It's called the constant region, and all humans have antibodies with the same constant region. All mice have antibodies with the same constant region, but their constant region is going to be different than the human's constant region. So I'm just going to shade in very sort of quickly, lightly, where the constant region is, and it's going to include my disulfide bonds. So I'm going to have to try and shade around them. But that's the constant region on one heavy chain, and then it's matched on the light chain. So I'm just going to finish this shading, and then I'll show you what I've got. So those are the constant regions, and that's going to make it the same for all of the antibodies of a species. The rest of it, this unshaded part, that's going to be my variable regions, and those are going to have three different sections, two different sections depending on a heavy or light chain, and those are coded for by different sections of DNA. And the B cells, when they are making their mRNA sequence, they are like randomly selecting which sections they're going to use. So the first section that we'll talk about is called the V, or the variable segments, and that is at the top of your heavy chain. So you've got a variable segment there. I'm coloring it in red. You can use whatever color you want, but it needs to be the same on both sides. So this is going to be my variable segment, and there are 51, I think, different variable segments that you could have. So the B cell is going to pick one of those 51 segments, and that's what it's going to use to build this portion of the antibody. They next have a D, diversity segment, and I want to say there are 26 different diversity segments. So I'm going to use blue, and I'm going to shade that in. So this section here is the diversity segment. And then on the heavy chain, you're also going to have, last but not least, a joining segment. So this is a J segment, and I want to say that there are 6 different options that the B cell could choose for its joining segment. So those are just the variations that are on the heavy chain. The light chain also has variations. They have 2 different variations. They have the variable segment at the top, and then I haven't used purple yet, so that's green. And then they also have a joining segment, which is towards the bottom of the light chain. So I'm going to color that in purple, and I realize that it's a little bit hard to tell the difference between the purple and the black, but I will label these in just a second. So there are now 5 different segments, and I used certain colors to represent the differences there. Maybe you picked different colors. That would be totally fine because they're coding for different amino acid sequences, and therefore they're going to make a slightly different shape. That shape is really important, but let's label these first. So the first segment are J, and that is wrong. The first segments are V, J is at the bottom. So my V segments are at the top of both the heavy and the light. So it goes V. Then only the heavy chain has the diversity D segment. But I do have a J joining segment on both the heavy and the light strands. So those are my segments there. And the reason that this is important is because the antibodies have to be able to attach to antigens, and they attach based on their shape. This is why protein folding is so important, is because if it folds it into the right shape, then it's able to do its job. And the amino acid sequences, those are controlling the shape. So the place where my antibody can bind to an antigen is right here at the top. So I'm going to just draw in a antigen. And my antigen is going to fit sort of like that. And it fits based on these different colored regions. So these different colored regions are going to make it a different shape, and that different shape is going to make it so that it fits with a different antigen. So down here, I'm going to just label that this is an antibody. Whereas this purple thing at the top, that's an antigen. And then the black portion of my antibody is called the constant region. And then I'm not going to do any more labeling on the top. So this is an antibody. It connects to an antigen. We're going to make it now using pipe cleaners. So you are going to need 2 pipe cleaners that are the same color and the same size. So 2 pipe cleaners that look like this. You are going to need 1 pipe cleaner that's a different color that's cut in half. That's going to be our light chains. And then you're going to need 1 sparkly pipe cleaner that is cut into 4 pieces. So I'm going to give you a second to get those, and then we'll move on. Okay, now that you have your pipe cleaners, we need to get these all put together. So my yellow ones are my big ones. Those are going to make my heavy chains, and so this is going to make my basic Y structure. So go ahead and bend those about halfway down so that they make a nice Y. And those are held together by 2 disulfide bonds. So I'm going to use my sparkly pipe cleaner, and I'm going to make 2 disulfide bonds. So there's 1 disulfide bond, and then a little bit lower, I'm going to put my other disulfide bond. And those are the heavy chains. The heavy chains have light chains attached to the side of them, also attached with a disulfide bond. So go ahead and use your sparkly pipe cleaner to kind of make those stick together. And do that on both sides. The next thing that you're going to need are some beads, and the beads are going to represent our variable regions. So our variable, our diversity, and our joining regions. So for this, you're going to need random colors, but you need 2 of each color. There are 5 different sections, so you need 5 colors, but you need 2 of each color that you choose. So go ahead and pull out whatever colors you like. Just make sure that you do have 2 of each. All right, so I've got my 5 segments. We're going to start with the heavy chain, and I'm going to pick one of my bead colors, and it's on both heavy chains. So I'm going to put those ones opposite each other. So you can see there that I've got 2 pink beads. They do not show up very well, but they are there. And then I'm going to put my next, which is my D region. So I'm going to add another bead. This time I've chosen black. That one's much easier to see, kind of. And then pick your last region. This is your variable region. This one has the most different options. And add that, and now I have pink, black, red. And you probably have a different order because you chose randomly. Now on my light chains, I've got 2 regions. So I'm going to put yellow first, and you're doing this randomly. And then blue is my variable region on the light chain. And now I have an antibody. And what I want you to do is I want you to look around the room. We did not have that many different colors of beads. In reality, we should have had 51 plus 26 plus 6-ish different colored beads. We didn't. We maybe have, I don't know, 15 different colors. But even still, it should be that when you look at the person next to you, you've probably chosen different colors. And even if you didn't choose different colors, you've probably chosen a different order. And if you have different colors or a different order, or both, you have different antibodies which are going to bind to different antigens. And that's all. One more thing. Please do not leave beads all over my classroom. You can take your antibodies home. Those are yours. You can throw them away. I don't care. If you want to throw them away, if you could put the beads back into the containers, that would be great so that I can just have extras of those. Otherwise, take them home, put all beads back into the containers, and clean up my classroom.